Okay, good morning, everyone, and welcome to the last seminar of the series of ELIXIR CZ Friday Coffee, which we present the tools and services which are provided by the infrastructure. Today, we have opportunity to hear something about CalFitter, which is web server for analysis of protein thermal denaturation data, created by Stan Mazurenko from Faculty of I'm sorry, it's of Nusa, which is I think Hospital of Santa Ana in Brno. So Stan, the floor is yours, and please give us the lecture.
Thank you, Eugene. Yeah, so I will introduce a little bit our group, so no problem with this. Yeah, today, thank you all for coming, and thank you for inviting me to give this talk. CalFitter web server, it was created in Loschmidt Laboratories. I'm not the only person responsible for it. Let me change the slides. Yeah, the slides are changing now. You have heard already several presentations from our group, and this is protein engineering group. So we are a part of FNUSA ICRC, but also Masaryk University. And typically, people in our group develop a lot of different tools to assist bioinformaticians, to assist people working on proteins, enzymes in all domains, but CalFitter a little bit stands out from other tools, primarily because it is aimed at experimentalists, not people with some programming background or some computer science background, but people who actually go to the lab and perform some measurements. Yeah, so it's a much more challenging group of target users. So just in a nutshell, proteins that we are working with, yeah, they are the strings of amino acids that are typically folded. And here in the middle of the slide, you see some examples of folds of proteins, and they are the main machinery of living organisms. So this proper folding is critical for their functions. And here on the slide, I just put a few interesting applications that even we are looking at in our group. So those are biopharma, of course, industry. A lot of enzymes are used in different steps of food processing, manufacturing, processing of different fabrics, colors, etc. And there are many, many applications. Biosensing, because we work, for instance, with proteins that can degrade toxic compounds. So then those proteins can be used as indicators whether those toxic compounds are present in the soil or not. But also interesting for us is mechanisms behind different human diseases. And it turns out that quite a lot of diseases are based on proteins misfunctioning in human bodies. And it's a target for biopharma, for therapeutics, but also it's interesting for us just from the fundamental point of view to understand what's happening, what's the mechanism behind this malfunctioning. Which brings me to protein stability, and this is the goal of the CalFitter, to help with analysis of protein stability. So in all those applications that I show on the slide, we work in a quite artificial environment for proteins. So then we typically want to have them stable in some non-native conditions. And by stability, we mean that they are properly folded, basically. So why is it important? First of all, if protein is properly folded, then it can withstand higher temperatures. So then if you use it in some industry, in manufacturing, and you need to have your processes at higher temperatures, that you want to have like functioning protein at those temperatures, then there might be also some very critical conditions, such as very high or very low pH, or presence of some agents that denature proteins, etc. So then proteins will lose their functions very quickly unless they are stable. Yeah, so then there are a lot of tools that are aimed at increasing stability. Another point why we want to have stable proteins, it's not even to preserve their function, but actually to drive chemistry to higher rates, chemical rates, because if you increase temperature and your enzyme is still properly folded, then typically all the chemistry happens faster. So then even without denaturing your protein, you can still improve the processes that you apply in your pipeline. Then eventually, people, when they perform protein engineering, so here on the upper right corner, we have some mutations. I try to represent mutations in the proteins. So that's what people in our lab do quite often. Yeah, they say like, well, okay, we have an enzyme, it's good, it works against toxic compounds, for instance, it degrades. Can we change this enzyme somehow? Can we replace some amino acids to make sure that it's even more efficient in degrading this compound, for instance? But the problem is that quite often when you introduce even a single mutation, you can completely disrupt your protein. Completely meaning that it will misfold, it will be highly unstable, it will be degraded in the cell, and you won't be even able to produce it. And this is just the effect of one single mutation. So then if you manage to first stabilize your protein, then you can play around with different other properties that you might be interested in, such as activity, for instance, with this reserve to spare, to use in stability. And then finally, I mentioned that human diseases are quite often interlinked with some malfunctioning proteins, and there is a big field of study, these single nucleotide polymorphisms, that even a single mutation in the protein, when it disrupts protein stability, quite often it then leads to some very unpleasant diseases in people. It might be anything from the protein not being able to signal or not being able to transport something in human blood, or start to aggregate and accumulate in a human brain, for instance, and then cause some really bad symptoms. So then it's also interesting for us to understand those mechanisms, and to do that, we need to measure protein stability. Speaking of which, so then if we look at the landscape, so here it's an easy representation of what's happening when a protein molecule is synthesized. So you have on the left-hand side, so it's synthesized usually as a string. And then there is a protein energy landscape, Gibbs free energy landscape, that is depicted on the slide. And then there is a minima, typically, that represents a functionally properly folded protein. But then, of course, there might be some local minimas, for instance, when there are some intermediate states, when the protein is not fully folded yet, but can still linger in this state. So typically, the biological like the synthesis happens from left to right. So you fold the protein. I mean, the cells, for instance, in the cells, the proteins are folded when they are synthesized. But of course, when we study, it's very difficult to just observe this folding process. Difficult in experiments, including also difficult in silico even. Yeah, if you perform some molecular dynamic simulations, sometimes you will have to simulate for in microsecond scales to observe some folding events, which is too long and it's beyond our computational capabilities. Of course, there are some ways how to do that, but the alternative approach is to go for experimental techniques where you denature your protein. And protein denaturation is typically that you increase temperature or you add some denaturing agent, and actually you follow the reverse process when you have a folded protein and then it starts to unfold. And then if you measure some signal, you can get some ideas, estimates about the differences in Gibbs free energies between different states, for instance. So you get some quantification of those states. So actually the process of denaturing, you can, like for those of you who are not familiar with protein engineering or biology in general, it's like when you heat up an egg. So first it is transparent because all the proteins there are more or less properly folded. But as you heat it up, those proteins start to unfold, they start to aggregate, they clump together, so you get this like white color, and then eventually you get your egg cooked. And then actually it's like any cooking process. When you cook meat, it changes its color also primarily because proteins become denatured. So when we want to study this Gibbs free energy landscape, of course, here on the slides, it's a very simple and easy to follow, I guess. But actually, in reality, it's a very complicated, so it's a schematic drawing. Yeah, we have very little understanding of how those energy landscapes look like. But what we can do is we can describe this process of folding or unfolding, which are just two sides of the same coin, with simpler mechanisms. For instance, mechanisms here at the bottom that involve some macrostates. So typically N stands for a native state. So this is functionally a folded protein, yeah, that is like at the bottom. Then we have intermediate state that might be like this state on the left or on the right. And then you have denatured state that can represent something at the top, for instance. So then it's a lower resolution description of this folding or unfolding of proteins, because now we have some, for instance, in this scheme, 3 distinct states, and then we can already measure the rates of transition between different states, how those, which states are more energetically favorable or less energetically favorable, or what happens if you introduce a mutation, what happens to this mechanism just in this very low resolution of macro states. So then once we do that, once we take on this framework, what happens when people want to measure protein stability? As I told you, in general, there are some high resolution techniques, but they are super demanding and they have a lot of different limitations by protein size, or that you need to go to France and book some slot. So people, labs all over the world, that they usually use much simpler methods. For instance, differential scanning calorimetry. So here on the left, what happens is the measurement, the machine records the heat flow to a sample. So you take a protein, you put it in a cuvette, and then you start increasing temperature by 1 degree of Celsius, for instance, and then you measure how much temperature, how much energy it consumes. And then it turns out that when there is an unfolding event, you will have a peak on this thermogram because protein will consume a lot of energy just to change its conformation rather than increase temperature. So that's why you have to, the machine has to send more heat to change, to keep up with the change of the scan rate, of the temperature. And this is how usually people measure. They say like, well, okay, let's pick the peak. The peak temperature, and this will be the melting temperature. So this is the only number that quantifies how stable a particular protein is. Then there are other techniques. They look a little bit different. Horizontally, we have the same x-axis. Yeah, we have just temperature, but vertically we measure, for instance, a fluorescence signal. And here the idea is when there is a transition region, you can see here in the middle, it means that when the protein starts to unfold, it exposes different residues to the environment that didn't used to be there because they were buried. So then you see the difference in fluorescence signal or absorbance signal, or there are different spectroscopic techniques. But the type of the data that people collect are very similar. And then again, they typically pick the middle point of transition. And then there is a third technique, which is a different x-axis now. So now what happens is you take a sample, protein sample, and then you heat it up rapidly to, for instance, 64 degrees or 70 degrees. And then you trace the relaxation, what happens as time passes. And then again, since it's high temperature, some molecules are still folded, but more and more of them start to unfold, and then you detect the signal, and then you can measure it. So here the challenge is those 3 techniques are very widely used. They are simple. Of course, the machinery is expensive, but this is a classical protein characterization technique. So whenever you produce protein, most likely people in the lab will always go and measure the protein stability in one of those techniques. And then they will report melting temperature, for instance, or how long a protein survives at a particular temperature. Now, the problem is that you need to analyze this data. And as I said, that picking maximum temperature might not be the good representation, because even in this upper left corner, you see 3 curves, but they were actually curves from the same protein. But what happened was that people just used a different scan rates. So here the scan rate of 0.5, 1, and 2. And you see that data actually resulted in a change in melting temperature, like the peak shifted by approximately 2 degrees of Celsius. So then the question is, okay, which temperature is then the melting temperature of this particular protein? And this is important, yeah, because when we stabilize the protein, we introduce mutation, then we measure melting temperature, we say, okay, we now have a protein that is like 3 degrees more stable. But actually here, which temperature to choose? Also on the right-hand side, it's like really strange, like how do you select this middle point of transition? What if there are 2 transitions actually, and you don't observe them in the data? And then eventually you have 3 different types of signal, and they might not agree because the underlying process, which is the same, yeah, it's just protein unfolding, and you expose it to high temperature, so it's even the setup is the same, yeah, it's just the temperature shock to a protein. But actually, you get 3 different sides of the same process, and then you want to have a tool to put them all together. And unfortunately, when we started working on this type of data 3 years ago, we realized that there was no such tool. There were some tools for individual analysis, but they included very simple models of just one transition, for instance, and for our proteins they didn't work. So then what started just like as a script in MATLAB, eventually we ended up with a server which is now called CalFitter, and now we're working on the second version of this server. Yeah. So the goal of the server is to allow people who collect those types of data to then analyze them, to quantify them in terms of Gibbs free energies, in terms of mechanism of unfolding, in terms of melting temperatures of proteins. So like get as much as possible from the data that people collect. So the workflow, the backend of the web server is as follows. First, the user usually selects a mechanism, and I showed you a mechanism on the left, and that was like usually the simplest, one of the simplest ones. But of course, you can have a very much more complicated mechanism. And nowadays, in CalFitter, I think we have 13 or 14 different models. So those are just models with different number of intermediates and different switchings between those intermediates. So then once the user selected this, the mechanism that they would like to test against their data, CalFitter performs a modeling of a dynamical system. So usually you can write down the system of differential equations that describe how each individual species will change in this scheme as a function of rates, and then those rates are functions of Gibbs free energies and melting temperatures. Yeah, so here in this system, there are lots of parameters hidden in the kinetic rates. And then CalFitter integrates those rates, and then what it performs is just fitting. Yeah, so I guess many of you are familiar with curve fitting. It's just for a different set of parameters, you generate like some line. Here it's a straight line, for instance, that I took from the internet, like this red line. And then you have your real data, and those are blue data points. And then you just try to draw this line through the cloud of data points. And on the left-hand side, typically you have some error that is then minimized. And by doing so, you select the optimal parameters that eventually fit all your data sets. So here it's a little bit more user-friendly workflow of the CalFitter. So the users will upload the data, then perform the model selection, they simulate initial simulations, then they perform the fitting, and then they can export those results. And in a moment, we will see how it happens. Yeah, so what kind of results they usually get. So now I think I have some time, and I will try to show you the CalFitter as it is. Right now. So it's like a web server now. We published the paper in 2018. There are two regimes, but now I'm focusing on this global feed. So you see there are three types of different data sets that the users can select. So once the users click on it, it allows them to upload three different types of data. Yeah, I will just upload the example that we have. I hope it will work. So here we will have three data sets for colorimetry, many different data sets for spectroscopy, and then also for kinetic studies for all those three types of data sets that I showed you. Yeah, and then the users will also need to set up some experimental settings, which I won't stop on now. And then they proceed to the next screen, which actually allows them to perform this global feed and model selection. So here there are multiple panels. The graph panel show different types of data. So this is the colorimetry data, for instance, and you see that here the profile is much more complicated than I just showed you. So it's not just like one peak, but there is like one peak, something in between, some negative peak. So it's like, and most proteins actually have quite sophisticated thermograms. Yeah, then you have spectroscopy. You see that some of them can have different scales, different units, etc. And then the kinetics, the relaxation rates. So then at the bottom, there is something that is called the panel called data pretreatment, where people can change their data. For instance, for spectroscopy, we now see that they are all not scaled. So we can just by clicking on one checkbox normalize all the data sets to one scale. Yeah, people can also cut the data because you see that there are, for instance, some artifacts and which is quite common in experiments. Even if you take the same protein, the same batch, the same person goes to the same lab and just performs 2 measurements in a row, then one measurement is good, the second measurement was bad because something started to stick, for instance, to some cell walls, and then the signal didn't go through. So then this panel allows people to play around with the data and just select, deselect some sets and also rescale them. And then finally, people can select those models. Once they select a model, 2 panels appear, for instance, here for 2 transitions. And then immediately CalFitter will try to simulate all the signals for all 3 data types based on the parameters that appear in the window, which is why it's quite more involved process than just so unprepared user cannot just like go to this web server and just like click through it, perform some automated fitting, and then get the results, because it will require a lot of manipulation and understanding your data, understanding different parameters. But it turns out that quite a lot of groups, they actually look deeper into protein denaturation, so they have some meaning for those parameters. But in any case, so you see that here, for instance, some data sets are already nicely fit in, even with the starting points, but some data sets are really, really off. Yeah, then the kinetics is also not matched completely. So now here we have 2 steps, so there is some state decomposition that we can just remove from the graphs. And then what the user can do, they just press on the feed button, and then the CalFitter will try to improve the match between the model signal and the data signal. Yeah, so I'm not sure if it will be successful or not, because I just picked one model at a time. But typically, yeah, so you see that some parameters got updated. And for instance, for kinetics, we had much better fit. For thermodynamics, for calorimetry, we have now peaks much closer. But for instance, this part for high temperatures is not explained by our data. Yeah, and then here, well, we already started with a good fit, so then it's fine. There are also some statistics that, again, sometimes people make decisions based on this. Sometimes it requires some analysis, but what people can also do, they can download all this session in a run archive in like those small images. They can also download raw data and even those blue lines, so they can get access to like all the data that was used to generate those images, for instance, for their publications. Then typically for our user tools, there is also job ID that people can use to access the data later on. But this is basically it. So then this tool just allows users switch between different models, play with parameters, see how they affect the simulated signal, and compare this signal with their experimental data. And then eventually they can have a much better understanding of the process of unfolding than just selecting peak temperature, which I showed you. It's not that robust, and it's typically quite a lot of people who work on protein stability, especially from computer science part, they don't know that many experimentalists, they just take this peak temperature, for instance, and then they just report it in the database, which, as I showed you now, it's a very prone to error methods of evaluating. Yeah. So now, yeah, we will skip this interface. So the expected outcome from this the session, after people play around with the data, they usually have a mechanism of unfolding, so they understand how many intermediates there are, for instance, on the unfolding pathway. Does it happen just like immediately, or there is some semi-folded state, which is important because you see here at the bottom the same 3 lines with the DSC, and I already told you that if you have different scan rates, you might have shift in melting temperatures. But here the situation was even worse because there was one more transition that is not visible as a separate peak, but actually it happens at lower temperatures over here in blue line, which we could understand from the fitting. And this is important because when you measure protein stability, you don't care much about this just apparent melting temperature. You care much about what is the temperature when you lose your function. And it might be that you actually already lose your function after this blue transition that happens at like in the very beginning of the peak. So then you will be over evaluating the melting temperature of your protein and the stability of your protein compared to the real data. And then of course it's a proper Gibbs free energy estimate, so you get a more robust delta G estimates than what usually people report in publications, because what they report, they use built-in software in this measurement tools, which has a just very simple model. So as I told you, even for our proteins, it already didn't work, and for many people, it might also be not the best solution. Yeah, so here we already had a few case studies, like nice graphs. So here you see different types of data in different colors, and then black lines are the fitting generated by CalFitter, and then at the bottom you can also understand how fractions of states change when you change the temperature. Yeah, so the summary, yeah, it's a very complicated process of protein unfolding. We now have the tool for analysis of it. Over the 3 years, we had more than 4,000 visitors and already 20 citations, which might seem modest, but you need to take into account that this requires a lot of biophysical expertise, yeah, this kind of analysis. So actually more and more people are starting to use CalFitter, and for us, of course, it's very great that we can give back to the community. And now we're actively working on CalFitter 2, so stay tuned. We will update and improve some functionality and add some new types of analysis to our CalFitter. So this is all on my part. I hope you were able to follow it, and I'm happy to take any questions.
Okay, Stan, thank you very much. It's a really interesting tool. And we already have 2 questions. So first is, does your tools take into the account reversible versus irreversible denaturation? I think it's from Wojta.
Yeah, it's actually a great question. Thank you. And actually, that was one of the motivations for the development of the CalFitter, because our proteins, when we collected the experimental data, we saw that they were irreversibly denaturing. And there was no tool to analyze irreversible denaturation, no tool to perform the measurement. So we had to write our own scripts, basically, in MATLAB, then Python, and that's how the idea for CalFitter came about. So now, out of those 13 models, we have all types of combinations. So there are like first step reversible, first step irreversible, second step might be reversible or irreversible. Yeah, like we try to cover as many models as possible. Ideally, we would also want to have some sort of editor where people can in turn insert their own scheme. But this is a challenging part, so we are not there yet.
Okay. My question is, you can upload your own data, yeah, and get the analysis. So is there any requirement for quality of the data? Can you, for example, upload a CD spectroscopy data for the denaturation? It's ready to fit it even though?
What do you mean already fitted? So you mean like already processed, pre-processed, yeah?
Yeah. So have you any requirement for the format of the data or the quality of the data? How you...
So yeah, in terms of format, now version that we have now, it allows uploading comma-separated files, and it's up to user what kind of data they would like to upload. So for instance, when they collect CD spectra, they typically choose a wavelength, for instance, specific wavelength, and then they upload only this particular wavelength data to CalFitter. Yeah. Now we work on this extending more to allow uploading the whole spectra, like in different wavelengths even. And so this will be the second version of the CalFitter. In terms of data quality, it's really on the user side. So CalFitter will take anything and it will try to fit anything. And we also implemented some correction, because you might have 2 different types of data, one with a lot of noise, for instance, and one with a very small noise distribution or many data points. So actually CalFitter, since it fits globally into those data sets, it also reweights different measurements based on the error that is present in the data. Yeah, so it can handle quite flexible types of data.
It's very useful. The last question, how many visitors or how many users you already have for your CalFitter?
Yeah, so now it's like 4,000. I think the statistics is on the side. Let's see, yeah, because if we go, so there are around 4,000 visitors, around 3,700 jobs already executed, which is quite funny. But actually, it's quite often you have many people working on the same data set. Yeah, because again, different techniques. So then you might have different users just accessing the same data set. And as I said, we already are like the number of citations is increasing, and this is partially why we are motivated to develop it further, because we see that there is demand from community, and we got some feedback from users also whenever they had some trouble. They said like, oh, thank you very much, because this irreversible transition that Wojtek mentioned, it's pain for many researchers, that they need to then invite someone from like with computer science background, programming skills, to write the fitting procedure. But here they can do that without any programming experience. Yeah, they just upload their data and select models and play with it.
Okay, Stan, thank you very much for your lecture. Thank you all for listening, and we will see each other next year for the next Friday Coffee. So goodbye and
goodbye.
Have a good day.
Have a nice New Year.
Bye-bye. Have a New Year.